All right, we are live. Start off with Tyler, and he's got number 1.
All right, so number 1 is talking about the genome of different viruses. So for number 1, we came to the conclusion that it was a circular RNA double-stranded virus to the percentages of the different and that is resistant to exonucleases. For genome number 2, genome of virus number 2, we came up with RNA single-stranded virus, and due also the percentages both to exonucleases and endonucleases. For genome number 3, came to the conclusion that it was a linear DNA double-stranded virus. And then for genome of virus 4, we came up with it is circular DNA and single-stranded.
Sweet. So then
it's me.
Eliza's got number 2.
Okay, so the first one, it's either topoisomerase or DNA gyrase. Topoisomerase 2 is composed of 4 protein subunits and uses ATP to compact bacterial chromosomes, while a nucleosome contains 146, 107 base pairs of DNA and 8 histone proteins. Double helix is 2 nanometers wide, and adjacent base pairs are separated by 0.34 nanometers. DNA double helix. Topoisomerase 1 is the one that introduces positive supercoiling and cuts a DNA strand. Origin of replication E. coli is ORI.
Looks like we lost Eliza.
Oh, no.
Oh, no.
Just having all kinds of issues tonight.
I know. So she was just about to talk about the replication origins called the
What's that?
There you are. Eliza, you're back.
Sorry, I'm at the library and the thing came over the intercom. Okay, at public library. So, sugar in uridine is ribose, and in sugar in deoxythymidine is deoxyribose. Okay, that's me.
All right, on to number 3 with Sky's gonna, or no, Sophia's doing number 3.
Okay.
So, Sophia, whenever.
I just got a message saying she can't hear us.
Yeah. I sent her a text. Just tell her to start talking about number 3 because I think we can hear her.
Yeah, I was able to hear her.
Yeah. I think so. So Sophia's got number 3. Let's try and Sky and Tyler, can you mute your lines real quick?
Yeah.
Thanks.
Yep.
All right, her video is lagging. Here are the answers to number 3. I'll make an addition to this video, and I'll have her explain that in an additional video. We'll move on to number 4, and
all right.
Sky will explain that for us.
So I have number 4, and it says the bacterium, Macrobacterium tuberculosis, has single circular chromosome that is 4,410,000 base pairs in length. And it says if the chromosome was stretched out linearly, how long would it be in micrometers? So basically to do that, you take the 4,410,000 base pairs, and you just have to convert it to micrometers. And so one base pair is 0.34 nanometers, and then 1 micrometer, or yeah, 1 micrometer is 1 times 10 to the 6, or 1, yeah, 1 times 10 to the 6 micrometers, and you end up with 1,499.4 micrometers. What is the diameter of the M tuberculosis chromosome in micrometers? Note circumference is equal to pi times diameter. And basically, it's pretty easy. You take that micrometers and divide it by pi and get 477.3. Then C, suppose that M tuberculosis genome is organized into loop domains. Each domain contains 50,000 base pairs. If one of these loops was stretched out linearly, how long would it be in micrometers? It ends up being 17 micrometers, which, I mean, you can see the work there on the screen. And then number D is how many histones are found attached to the M tuberculosis chromosome. Ends up being 0 histones because histones are not found in prokaryotes, but only in eukaryotes.
Yep. And then on to number 5, which is mine. So we're just going to answer these questions real quick as they give these different figures. So it says, which of the molecules are nucleosides? A nucleoside is a molecule that doesn't have a phospho group on it. And so we can look at this, and A, no phospho group, B, C, and D all have phosphate groups on them, and then E and F do not have the phosphate groups. So that's A, E, and F. And then the nucleotides are ones with phosphate groups, so that would only be B, C, and D. Makes that easy for us. Then which of the molecules contain the ribose? And so ribose would be the ones with the oxygen, or with the extra hydrogen right here, our alcohol group. And so the alcohol group F, E, no alcohol group on D. C has the alcohol group, and then A and B do not have the alcohol group. So your answers are C, D, and F, or C, E, echo, and F. And then which ones are deoxyribose? Are the ones without alcohol groups on there. So that would be D, A, and B right here in the 2 position. That's where those alcohols would go. And then which molecules contain pyrimidines? And the trick with this one is purines and pyrimidines are different. And purines, the trick that I learned, purines are pure. They're pure as gold. And pure as gold, gold wedding rings. And so it takes 2 wedding rings to make a marriage. And so these are purines with the 2. And that means everything with the single ring is a pyrimidine. So C, D, E, and F. And that's pyrimidines. So that is our video for group 3 today. We'll make an additional one for Sophie so you can explain that problem 3 for us. And thanks so very, very much. Get everyone's faces there at the end. And see you next week.